Today we have a guest speaker who came all the way from the South Bay. She came from Stanford and is a wonderful speaker. She came last year as well. I think you're really going to enjoy it. And her topic is bioluminescence imaging. Do you guys remember what bioluminescence is? What is it? Chemical reaction. Doing what? Producing light. Good, good. Okay. And how is that different from fluorescence? What's the difference there? Fluorescence is chemical. Yes. Fluorescence, you put in high energy light and it gives you low energy light out. Is it a chemical reaction, fluorescence? No. Okay. Bioluminescence is. Okay. So our speaker is Dr. Jen Prescher, and she is a postdoctoral fellow. I think she's going to tell you a little bit at the beginning of what it takes to become a postdoctoral fellow. And she's very nice. Feel free to ask her questions. I'm sure she would love to talk to you guys about it. But you're going to get to see a lot of really nice images today. Okay, so she will have to turn off the lights at some point so you can make sure to see things well. With that said, I'm going to turn it over to Jen. And thank you.
Sure, you're welcome. All right, it's great to be back and share with you what I think is one of the coolest natural phenomenon found on the planet, and then how we've sort of taken this concept of bioluminescence and using chemistry to make light for purposes in medicine and understanding biological systems. So, as Marco said, feel free to interrupt me anytime if you have any questions or if something doesn't quite make sense. And on this first slide, I'm showing you an example of bioluminescence that some of you may have actually seen out in the ocean at night. You can often see some bacterial species giving off light as waves crash on the shore. But before we talk a little bit about science, I'm just going to tell you a little bit about myself and my scientific career path. I think it's always fun to give a little background on the speaker at the start. I don't know if it's... Can you see this okay? I'm originally from Minnesota, in a small little farming community there. I grew up on a farm about 2 hours southwest of Minneapolis, and there I grew to love sports, of course, and unfortunately the Minnesota Vikings are perpetually horrible, but I still like them anyway, and I have a lot of fun still playing out in the snow and playing tennis. And after growing up in Minnesota, I completed my undergraduate work in Wisconsin at the University of Wisconsin, their campus in La Crosse, which is just right on the Minnesota border there. And this is where I got involved in a little bit of undergraduate research, which some of you may or may not contemplate doing. And I was interested in learning how to make molecules and organic chemistry. And this is a picture of one of the hoods that I use. They're sort of all cluttered up with all sorts of glassware and gadgets, and this is me back in the day doing something you should never repeat as a chemist, the goggles not over my eyes. And then from Wisconsin, after deciding that research was sort of for me, I moved on to Berkeley here in California to work on a doctorate degree in chemistry with an emphasis in chemical biology. And how I like to describe my thesis research there is that it kind of involves the study of peanut M&Ms. You know, when you slice a peanut M&M in half, you get the peanut in the middle, which are kind of like proteins, right? And then surrounded by that layer of chocolate or fat, and then the outside is that candy shell or sugar. And it was the sugars that I was interested in studying at Cal. And this is an actual cell right here, or the peanut M&M with all the proteins in the middle and the lipids on the outside. And then this kind of gray haze region is the sugar coat of a cell, kind of these little obscure features. And I'm blowing up a region of that there where you can see these sugar units, or the more appropriate term for them on a cell is a glycan. And we were interested in studying how those particular structures change as a function of the health of the cell. So you often see these dramatic changes in glycan structure or sugar linkages in response to things like cancer and development, inflammation, and so forth. And so we developed a bunch of different small molecules to target those particular glycan structures, and then we also came up with a few nifty chemical reactions that could be used inside living animals to look at those structures. And then since completing that work at Berkeley, I moved on to Stanford to follow up on this a little bit, and not only being able to target those particular features, but actually image those structures. And so I'm showing these like little stars here, and can we actually image how those sugars influence cell migration inside living animals? And then can we visualize these changes that occur with things like cancer and other physiological processes? And I've been at Stanford now for a little over a year, and I'm now moving on to become an assistant professor of chemistry. And so I'll kind of be taking all this previous like research into carbohydrates and imaging and looking at cells to be able to look at still more physiological processes inside living animals, which we'll talk a little bit more about today, including using bioluminescence imaging to track things like T cells, B cells, and all the different components of your immune system. And then not only being able to look at them inside live animals, but zoom all the way down to the vessel level inside these animals using fluorescence-based approaches to look at cells moving in and out of the bloodstream. So that, in a nutshell, is what's taken me from Minnesota all the way out to here, and then I'll be transitioning on to another position shortly. But today I'm going to talk to you mostly about one of these imaging techniques that I've used extensively at Stanford called bioluminescence. And we're just going to cover some of the cool things about bioluminescence and all these colors you can get out of animals and how we can use that then in medical applications, and the actual mechanisms and chemistries involved in light emission. And then right before the break, we'll also talk about how we actually in the lab take those natural glowing organisms, harness that light emitting power to look at cell tracking inside animals. And then after the break, we'll come back to looking at all the different things my research lab and my colleagues at Stanford have used this for, including looking at immune cells and cancer cells in vivo, as well as anything related to infectious disease. And then probably the most fun part is thinking about where do we go from here, right? There's all these cool things we have done, but so many unanswered questions related to this topic that you yourself might be interested in pursuing. So bioluminescence, well, we kind of talked a little bit about at the start with how does it relate to fluorescence or other light-emitting processes. All of these things involve just the simple excitation of an electron in a given material, and when that electron relaxes back to the ground state, light is given off. And all of these processes of light generation just differ in what is used for that excitation energy. So things like the sun and the light bulb and stuff, they use a thermal-based approach, right? You heat up the filament in the light bulb, and it gives off light and lots of heat as well. The light bulb gets hot. So these are kind of very inefficient processes of giving off light. And then what you probably talked about more recently is using photon excitation, or using lasers of light, or just light in general, to excite electrons and then get light out of the system. And kind of the classic examples, if you've ever taken a UV lamp and held it up to club soda, it starts fluorescing because of the fluorescent molecule quinine that's in that material. But today, the light emission process that we're going to talk about, which you had mentioned before, involves a chemical-based excitation, or using some chemical reactions to get out light. And everyone's probably familiar with glow sticks, of course. That's using the same process to give off light, a chemical reaction. However, in this case, most of these processes have to be super efficient at light generation, right? No energy can be dissipated as heat, otherwise you'd start burning your hands, right, with a glow stick, similar to if you held a candle, that wouldn't be very good. And the exact same procedure is involved in bioluminescence. It's set that you can see these images. They're naturally occurring systems of chemical reactions inside of things like jellyfish, bacteria, and other organisms that we'll talk about that emit light. And again, it's a very efficient process. Almost all the excitation energy coming from that chemical reaction, when the relaxation process takes place, almost all photons are released as light and not heat, right? Otherwise, the organism would burn up, or you burn your hand in a glow stick. And I think what's really fascinating in the ocean, for example, almost 90% of the things, marine species inside the ocean, are bioluminescent to a certain extent. And I'm showing you just some examples of krill and some other species of jellyfish. I'm going to turn this off for a little bit. Many examples in the sea. And there are a few examples on the land. Click beetle is the classic example, is the firefly, of course, that many have maybe seen in your backyard. I know in Minnesota there's tons and tons, and you can just grab them by hand out of the sky. I haven't seen too many fireflies in California. And then here's just a partial list of everything that we've identified thus far that produces bioluminescence. And the ones highlighted in yellow we'll talk a little bit more extensively about, bacteria, fireflies, click beetles. But probably the most striking thing, if you look at this list of all these different organisms, is most of the higher mammals don't give off light, right? Like humans, we don't walk around giving off light, or we don't bioluminesce. And that becomes critical for medical applications that, if we want to use light-emitting reactions to look at processes happening either in humans or small animals, that we don't have any light interference from our own bodies. There's no background signal here. And we'll talk more about that later. But within all of these different organisms that have evolved bioluminescence, it kind of makes sense why, certain animals, especially within the sea, would want to be able to emit light. Of course, one obvious reason is it serves as a little flashlight, right? So you can, have a little flashlight in deep sea looking around for food and things like that. Some species of fish use bioluminescence for that purpose. Others use it kind of as a brilliant display to attract their mate. And then other classic examples include using flashes of signal to scare off predators. That's what this shrimp here, that's vomiting bioluminescence into the face of a predator is attempting to do. There's some very elegant systems of fish that live together with bacteria in their stomachs. The bacteria give off light so that the underside of the fish is lit up. When sunlight hits that fish from the top, then it doesn't cast any shadow, right? That makes it very advantageous for that particular species to avoid being eaten by a predator. And I'm going to just tell you about a few of my favorite examples of bioluminescence. This particular species here is a dinoflagellate. It's just a fancy type of bacterium in the sea, and this is what's responsible for bioluminescence. You often see it crashing waves. They're a very common type of bacterium in the sea or the ocean, and they were responsible for that enormous amount of light in the wave that I had on the opening slide. And two other examples from the sea that are quite cool. This particular species of krill has different little pockets of luminescence down its underside that it kind of displays on and off, and the different flashing patterns or the different repeats of those lights help determine which species of female krill that male will mate with. And then this is a very striking example coming from this shark of how bioluminescence can be used to an advantage for food gathering purposes in the sea. What this particular shark does, its normal shadow is pictured here, but its underside is just filled with bioluminescent bacteria, which makes this shark look really small compared to anything floating underneath it, right? It's casting a very small shadow. So, larger fish come in thinking that that's like an easy prey for it, and it's able to latch on and kill things almost 10 times its own size just by kind of faking it out with how large and how potent of a predator it really is. And then another example of being able to trick other organisms around you into falling into their mouth to eat. Several species of anglerfish kind of co-opt bacterium, and they place it off of this like long rod off of their nose, and it's dangling out there in front of them, which kind of captures the curiosity of a lot of other fish. When they come near that, of course, there's a big surprise waiting for them. So I thought that was a cool example. And just to highlight that this isn't just random phenomena in the oceans going on all over, you can actually see bioluminescence from satellite images. And this image was taken just a few years ago, where this huge pocket of luminescent bacteria was spotted off of the Horn of Africa. And if you kind of lay it aside, it would stretch all the way from Monterey down to Santa Barbara. That's trillions and trillions and trillions of bacteria in there. Quite amazing. And it's the same. This is probably due to some of those dinoflagellates that I had up there earlier, the bacterium. And some sailors have even reported when they're lost at sea and they have a boat wake behind them, any agitation, dinoflagellates tend to luminesce when they're agitated, that they've been able to find their way back to shore or retrace their route by just following the wake of their boat and that trail of bioluminescence back to shore. So anyway, I think this is pretty phenomenal. You can actually see it from a satellite image. And in almost all cases of organisms that possess bioluminescence in the ocean, they give off visible light, which we can see, but it's always, blue in color, blue-greenish. And why do you think that is? Why don't you think we have, bioluminescent organisms in the ocean that are emitting, red light a lot? Any ideas on that?
Doesn't take as much energy.
Say that again?
Doesn't take as much energy.
It doesn't take as much energy. In what sense?
Maybe photons that they get only light in the blue region.
So the blue light actually has higher energy.
Yeah.
Think about where this might be occurring in the ocean. Yeah.
Maybe stand out too much before the light, so you might be concerned.
Yeah, so organisms living at the very depths of the ocean, let's say where a lot of light-emitting reactions are required, they don't have a lot of access to the sunlight, so they don't even know how to respond to lower energy red light, right? So the higher the energy of the wavelength, right, the farther it can travel. When you're way at the bottom of the ocean, what color of light do you think has the best chance of getting through? Blue, right? Or UV sorts of colors. And I'm just highlighting here, most bioluminescence that's occurring at the depths of the ocean, these organisms have evolved to respond to high energy forms of light, or what they're most likely to get a sense of if they're traveling up and down in the ocean. Blue light travels the best through water, so they've all been adapted to responding to blue forms of light. But if something came along that could sense something with red light, that would be a huge advantage for that particular fish, right? No other organisms would know how to respond to red light. They might not even be able to sense it. And we'll actually talk about a species of fish that's kind of on the evolutionary trail to responding to red light. But now, organisms on the land that emit light are more red-shifted in their emission. Fireflies tend to emit yellow light, if you've seen flashing bugs around. Some species of beetles have glowing green eyes. Worms sometimes can emit two different colors of light, and even some species of mushrooms, if you've been in the forest, if you look carefully, you'll notice them emitting green light. How many of you have ever seen glowing either mushrooms or fireflies or anything like that? Yeah. Are there a lot of species of mushrooms around here that do that? I haven't seen any mushrooms yet around here, but I've certainly seen some fireflies and some cliff beetles, and many different species of bacteria, although the colonies have to be pretty decent size, I think, to notice that sort of luminescence. And so in this case, why do you think that most of these organisms are emitting kind of yellowish colored light, yellow-red versus blue.
Sunlight?
Yeah, right. Other features of them have been engineered to respond to, right, their eyes can see red and yellow colors of light, and so the bioluminescent systems that evolved emit, green, yellow, more red colors of light. So the big question then, I'm a big chemistry buff, right? All my training's been in chemistry thus far. What's the chemical reaction that these organisms use, right? We can maybe take some cues from the reactions that are present in glow sticks. All of these animals are giving off different colors of light. That probably indicates that there's maybe at least a few types of chemical reactions going on to produce different colors. So if we take a look at the chemical reaction that involves light emission in the red glow stick, often before you crack it to give off light, there's hydrogen peroxide sitting in a little tube inside there, and that's what you crack to release. That reacts then with this funky looking oxaloacetate species, and then you end up forming some really, really strained molecules that are just dying to burst open at this bond. And when it bursts open, that's the energy that's transferred to, in the case of the red glow stick, a red dye sitting in there. Okay, and that puts that dye into an electronically excited state, and then the emission ends up being red in color. And so for a green glow stick, the only thing different in that tube is the color of the dye that's accepting the energy from this particular molecule. Now, for bioluminescence, we can probably rule out this chemical reaction occurring in most organisms because there's a large amount of hydrogen peroxide required for this particular reaction. Hydrogen peroxide is not very good for a living system. It oxidizes, everything in sight. And it also needs to be a very controlled process as well, right? You see, fireflies flashing, they can turn it on and turn it off, turn it on, turn it off, okay, versus kind of being on the whole time and then just slowly dying away. And I think the experiment that was done to try to figure out how bioluminescence is produced or what the chemical reaction is really cool because it's so simple. This was back in about the late 1800s, I'd say, when there was this researcher named Dubois, and he noticed that some species of clams that he was eating were glowing, and he was trying to figure out what that was from. So he ground them up with a mortar and pestle, and he noticed that when he kind of tried to extract whatever was in there giving off light, that he could do it with cold water, but over time, the signal went away. Okay? But then he also made the observation that if he tried to take that ground up clam and put it in hot water, there was no light emission. Okay, so something in there was heat sensitive. But when he dumped the two things together, whatever was in the hot water and whatever had dissipated in light emission, that he could regenerate the glow. Okay, so there were a couple of things that could be said about this. One being that it looks like there's more than one component involved here, right? If you can mix something from here and regenerate light emission, and also the fact that there's something heat sensitive, that makes sense, right? It's a biological system, you heat up proteins, they unfold. Those sorts of thoughts are running through Dubois' mind. And what he ended up doing was calling he didn't know what it was, but he just called this fraction luciferase. And he thought that that was probably the protein-containing fraction, because if you heated this up, the protein unfolded or no longer produced light emission, or you tore it up somehow, you ruined it. And he called this, whatever was extracted with the hot water, luciferin. Okay? And the reaction between luciferase and luciferin, in his mind, was what was creating the luminescence. And this theory actually stood the test of time. It ended up being that bioluminescence was produced from an enzyme and substrate interaction. The enzyme being the protein, and the substrate, the small molecule, being the luciferin. And it turns out all these biological systems, there's a bunch of other stuff sometimes that play in these reactions. If you need a source of oxygen, if you need other stuff in there, other cofactors to make the enzymes work. That kind of varies between different systems. But the observation was made back in the 1880s that since all these animals were given off different colors of light, that there were likely to be differences between whatever the luciferase and luciferin were, what the actual protein, what the actual substrate were, but that they would all operate by this same mechanism, an enzyme-substrate interaction involved in the chemical reaction to produce light. And so I'm just going to take you through a couple of those reactions because I think they're super cool. Enzyme-substrate chemistry or enzyme-substrate interactions are biological chemistry, right? Enzymatic reactions are life's chemistry, I guess you could call it. So in the firefly or the click beetle, most of the insects running around on land that can give off light, they tend to give off light, again, in this green-colored area, and they all use the same small molecule, which the generic term has sort of stuck for all these. They're all called luciferin. And luciferin, I'm showing you the structure right here. It's just kind of this weird-looking, multiple heterocycles all locked together. And what this reaction requires is ATP, which you might be familiar with from either biochemistry or biology classes, like nature's energy source, right? And that's used in this reaction. It's attached to this carboxylate, forming this very high energy intermediate. And this is all taking place within the luciferase active site. Okay, the active site of the enzyme is where this whole reaction is taking place. Now, when it's in this highly activated form, oxygen can add across this bond here, and then the whole thing kind of collapses on itself, giving you that original molecule but in an excited electronic state. And that's then the species that relaxes back and gives off the photon of light. And this reaction is so efficient that quantum yield, I'm not sure if you've talked about quantum yields, how efficient, how many, if one luciferin molecule goes into that active site, how many photons of light come out? It's like 98% efficient. Right? That's remarkable, but it's been evolved to do this process. Yeah?
So if this is so incredibly efficient, why do they instead use some weird chemical combination glow sticks instead of just copying what's in nature?
That's a good question. I think you actually can find glow sticks that have this system in it, but keep in mind though, when you have a protein that's involved in a glow stick, the protein's sort of finicky, right? It likes to be kept at a certain temperature. It's not easily shipped. It costs a lot to extract proteins in a pure form out of the firefly. So the reason why our glow sticks use all those chemical reactions is because it's much cheaper, right? And they're more robust. But that being said, this system has great utility in studying, living processes for sure. But you can actually buy all different sorts of luciferin little gimmicky things like bioluminescent beer, and things. We'll talk about some of them in a minute. But this process, what I want to say, the structure of this molecule gives insight into the color of light that's going to be given off, okay? Because the process used to generate this excited state substrate is similar to the excited state substrate you would generate with fluorescent light, okay? So the color of emission here is dictated by the structure of the emitting species. It's just that we got to this electronic excited state using a chemical reaction versus incident light like you would in fluorescence. Okay, so it's all the same once we get to this step. The emission process is just like fluorescence. It's just that we went about it a different way. We didn't pump light into the system. We just fed it an enzyme. Does that make sense? All right. Now, there's all sorts of cool chemical reactions that are at play with luciferin systems that kind of change the wavelengths of emission. So in many of like the jellyfish species and some of the bacteria, the color of light given off is blue, indicating that the molecule that goes into the enzymatic reaction is likely a totally different substrate. Because when it's electronically excited and giving off a blue wavelength of light, it should probably look a little bit different than the luciferin that was used by the firefly. So this is like a really bizarre structure in my opinion, and it's called cilantrazine. And this is actually the small molecule and the enzyme luciferase system that you'll find sometimes in different clams that you may have eaten. Every once in a while at a restaurant, a bioluminescent one is served, at just randomly cut it open and it's luminescing on your plate. So be on the lookout for that. But these are totally edible. And then the last, I think, reaction I wanted to show you is how all these different luciferase small molecule systems have evolved. Bacteria use really weird random aldehydes, like long chain aldehydes with a common cofactor that's found even in humans, one of the ribonucleotides. And after oxidation of that aldehyde, a blue photon of light is also given off. And I think the cool thing about bacteria is that they encode both the synthesis of their enzyme, luciferase, and they also encode the synthesis of the small molecule. For many of the other enzyme substrate systems, the luciferase-luciferin systems, those species are either eating it from somewhere in the environment or they're living together with bacteria that are producing the small molecule, and then let's say the fish has the luciferase, and they kind of work as a team to create their luminescence. But the bacteria, they figured out how to do most of that themselves. And then I'm not going to belabor the point, but there's so many, I think, interesting systems out there if you're like into small molecules or synthesizing things that exist in nature. The cilantrazine and the firefly system I've showed you already. But look at some of these like structures here. We've got something that looks really similar to chlorophyll, serves as the luciferin substrate in a enzyme substrate reaction used to generate light. And this thing here, I don't even know how to describe that, but it looks impossibly, or horrifically challenging to try to make something like that. But look at all the energy and the biosynthetic pathways that probably an organism invests in making these elaborate structures, for this purpose of giving off light. I think that's pretty incredible. But there's all sorts of things you can imagine that you can do with a luminescent system. In addition to what we talked about with firefly luciferase, luciferin glow sticks or something. There's been some exhibits, even one here in San Francisco, of like putting little luminescent bacteria in all sorts of colorful little patterns and letting them grow out into cool shapes, and then having a glow-in-the-dark exhibit of bioluminescent bacteria. As I mentioned before, a lot of these systems, especially in clams and stuff, people ingest normally. I've seen bioluminescent birthday cakes, and then if you extract enough of the luciferase and luciferin, they sell bioluminescent beer in Japan. I mean, all sorts of different things you can play with these systems. And there's even some places in the Caribbean where you can pay an extra amount to like go kayaking at night, right? You paddle around in your kayak and disturb some of those dinoflagellates, and you'll start to see them luminescing. If you haven't had the experience of seeing some bioluminescent organisms before, a deep sea dive even, or not even, it doesn't even have to be deep sea, but just a dive at night in Monterey Bay, you'll see flashes of light and things under the ocean. It's really, really cool if you haven't done that before. But it's always fun to actually think about, what else could you do with this? Some people have tried to make like bioluminescence, like Christmas trees, or things that just glow all the time as a replacement for like street lamps. I don't know if it will ever happen, but it's fun to think about. I don't know. Is there any other fun thing that we could use bioluminescence for? People? Bioluminescent people? Glow in the dark. It's like a code tag, maybe? Yeah. Keep track of kids at night or something. Anyway, but so if you see glow-in-the-dark Christmas trees or something like that, assume you heard it here first. But what I really came to talk you about today is how we've used this bioluminescent system, luciferase enzymes and luciferin small molecules, to look at biomedical processes and how it's facilitated research. And the simple point being that you all know, if you've held flashlights up to your hand and stuff, that light travels through tissue. Okay, so we have a perfect system here of something giving off light like a beacon. Luciferase and the small molecule luciferin in either a cell or in bacteria, we can use that to track things inside of living organisms. Because, like you said, right now, as humans, we don't give off light. So if there's light coming out of us, or if there's light coming out of a small animal, it's got to be due to the luciferase-luciferin system. It can be exquisitely sensitive because, right, we ourselves don't give off any light. All right, so that brings me to the second part of this presentation before the break, of how we can use that bioluminescence imaging system for this purpose of tracking things in vivo, and how we've actually taken that approach at Stanford. And the whole strategy for this approach is outlined here, and we'll come back to this a few times. So our thought process was, well, if we can use luciferase and luciferin together, either to used in bacteria or mammalian cancer cells, let's say, or encoded within viruses, we could use those in traditional animal models for either to monitor cell growth, cell location, you name it. Since those cells or viruses would be giving off light, if we just kept these animals and things in the dark, we'd be able to record where that light is coming from and get insight into the biological process that we are studying. And then at the end of the day, we'd be able to read out that light emission and watch how it changes over time. And so this is the whole strategy of using bioluminescence imaging. It fits into this larger genre of studies called molecular imaging. That is kind of a very popular term used in the press. And bioluminescence imaging is just one component of molecular imaging, and that is using any imaging technique, in this case bioluminescence-generated light, to track processes, to track cells, using some of these techniques inside of either mice or humans or you name it. And just to give you an example of how bioluminescence fits into the broader context of imaging techniques, it's no different really than things like PET, SPECT, MRI, a lot of clinical-based imaging approaches, like if you've ever needed to have an MRI done on your knee or something, or gone in for a SPECT CT at the clinic. This is a similar approach, bioluminescence, using some sort of light to interrogate tissues. And it's part of a whole class of different reactions being used at places like Stanford and research universities across the globe to gain insight into other types of biology. And the whole reason why imaging techniques such as bioluminescence or ultrasound or any one of those techniques on the previous page are very popular for biomedical research is that they allow you to look at things without perturbing the system under study, right? It would be better to look at cancer cell outgrowth inside the live animal versus just on, harvested random tissue. It preserves the actual context of what's going on inside that animal. And it also reduces, the number of living organisms that need to be used, because you can keep coming back to the same animal to study it. And you know exactly when things are occurring just based on what you're seeing. And you also gain a lot of insight into things you never really even knew were going on, that you could see, well, oh my goodness, all of a sudden this cancer has metastasized to this lymph node in response to this drug. You never would have known that before because you never would have known to look in other areas. And so it's become this remarkable research tool for discovery, kind of enabling advances in the clinic just by highlighting where different cells are and where different things are occurring. And I'm going to highlight once more how bioluminescence sort of fits in with all these other approaches to imaging, some that you've probably heard of just based on spending time in a hospital. Like CT, which is using high energy X-rays to blast tissue and provide information then on like bone density and other thick tissues. Bioluminescence, as we've seen, gives a readout of visible light. And so that's much preferred to a lot of other applications because it doesn't involve any like ionizing radiation, right? You don't have to wear a lead vest, with visible light, where we have visible light, on and around us all the time. So it kind of combines the best of both worlds in using very kind of middle of the road, lower energy sort of a readout, and it's highly, highly sensitive, okay, because of the fact that there is no background source of light energy, right? Mice and humans, we don't give off any light whatsoever, so you can see down to, far below the picomolar level, which is like on the order of a single cell, okay? That's really, really powerful. And we'll get into where bioluminescence becomes a great technique for use in animal research. But it's not perfect, okay? So as we'll discuss in a few minutes, visible light inside of either mice or humans is sometimes hard for that light to make its way out and be detected, and certain types of tissue are better for that than others, versus if you're using something like X-rays that are really high energy, they can blast through anything, okay? So you don't have any issues of, let's say, depth, or where is that light coming from, where is that particular cell at. So we'll talk a little bit about that, but for our research purposes, bioluminescence imaging is incredibly powerful. And the questions that we needed to address in establishing bioluminescence for use in animal imaging involved, how are we going to get luciferase and luciferin, the two components of our bioluminescent reaction, into other cells that we want to study? Like if I want to study human breast cancer, that doesn't have luciferase and luciferin combined with it. How am I going to get that packaged into a system so I can see those cells glow and then track them around? And then what sort of features need to go into a detection system so we can detect very low levels of light? That's kind of an engineering problem. We'll talk a little bit about that. And then we'll also talk about what actual bioluminescence images look like when they're coming out of this camera. But this is the basic setup. And we'll start first with looking at this kind of first question here. How do we get luciferase, the enzyme, into another cell of interest that we're looking at studying? If we don't just want to study fireflies for the rest of our life, how do we translate that system? And also the small molecule luciferin. How's that going to work? So introducing foreign proteins into cells is very common these days in biology and molecular biology classes. Some of you actually probably done some cloning as part of your coursework. The small molecule luciferin is actually free to diffuse almost anywhere throughout tissue. Just a small molecule, just like most of the metabolites in your body. It can go in and across the cell membrane, no problem. So the luciferin part maybe isn't as much as we're worried. Oxygen and ATP are certainly present in a living system, like a mouse, if we want to use that. It's the how do we get the luciferase in there, and that's where the cloning and molecular biology take place. So all protein, if we want luciferase in this cell, derives from messenger RNA, which eventually comes from genomic DNA. So we need to figure out how to get the luciferase gene from the firefly into the genome of our cells of interest. And how that would happen then is once the DNA would be transcribed in the nucleus to make the RNA and give us our protein, we can somehow latch the gene for luciferase in there, we end up producing proteins that have luciferase attached. And then if we just dump the small molecule luciferin on them, it's free to go in and outside of the cell. That produces then our glowing cell. There it is. Okay, so then the problem just boils down to what sorts of things can we attach the luciferase gene to? What do we normally use for, let's say, cell tracking? And we'll just take a quick diversion here to look at a gene up close. Okay, so every gene in the human genome and the mouse genome, they all sort of look like this. They have some stuff upstream of all of the DNA that's used to encode a protein, okay? And it's this stuff upstream, especially in this region here, the promoter, that becomes very important to us because this is the region within the nucleus of a cell where other things bind to and turn on the transcription of the gene. Okay, so if we want to turn on luciferase in these cells, this becomes the site that we vary, okay? Ultimately, we want to get this promoter region attached to the gene for luciferase. So whenever that promoter is turning things on, it's going to turn on luciferase at the same time. And just to be complete about this, when you actually express this gene all the way down into protein, a lot of these non-coding regions in the genome get spliced out, and the messenger RNA gets processed some more, and then ultimately the protein is produced. So what we want to have happen is whatever gene of interest we're interested in studying, if it's cancer-related, we want to use the same promoter region to attach luciferase to, so that if we're trying to study a particular process in cancer, luciferase turns on every time that particular gene turns on. Or if we don't care what the gene is and we just want the cell to glow all the time, then we just choose a promoter that's on all the time in a cell. Okay, we just swap it in and out. And that's sort of highlighted in this slide here. We actually make these synthetic gene constructs by simple molecular biology cloning techniques, if you're familiar with some of those processes. They become very modular. So I have, probably 20 different promoters, let's say I'm interested in studying, some related to cancer, some related to development, some just, that are always on in a cell. And I have many different luciferases sitting in my freezer, the genes. I just plug them in and out and make different constructs that I want to use for any given purpose. So we kind of just, like Legos, just kind of put them all together. And just to give you an example of what some of these are, you don't have to worry yourself about the names, but you may come across these at other points in your career of different promoter names that are always on in a cell. So if I always want to know where that cell is at any given time, I want it to be making luciferase all the time, I'm going to use one of these. Things like actin, which hold a cell together. That's always being produced, so I'm going to use that with luciferase if I always want to see where my cells are at. And we use these, of course, for any time we want to just do simple tracking experiments. But sometimes I only want the luciferase to be on and off in response to a certain event. Let's say when there's a huge inflammatory process going on. I want to know right then. So I'm going to put my luciferase behind an interferon gamma promoter, which goes through the roof when there's a huge inflammation taking place within inside the body. So in that case then, I only see the luciferase turn on when there's a huge inflammatory response taking place. So in this sense, I can have more control over what I'm looking at. The luciferase then becomes a readout of the biological event of interest. And this is what is classically referred to as a reporter gene. Okay, so luciferase is reporting on the event of interest. Does that make sense? Are there any questions about that?
So it's essentially like a light switch. So basically, this promoter is, when this promoter is activated, it lights up the luciferase, and when it's not activated, it doesn't light up.
Yep, it turns on its transcription.
Okay.
So the small molecule still needs to be there, but it turns on the transcription on and off, because that promoter is normally attached, let's say, to the protein of interest. So when that protein of interest turns on and I want to know about it, the promoter is what's controlling the transcription. So the promoter is going to turn on both the protein of interest and my luciferase at the same time. So yes, it's turning it on and off.
So then I just had a question about, like, when you have this, when you're transcribing these proteins, you make these proteins that are then activating your luciferase because you're making them luciferase as well. So what happens to the proteins when you stop making them? Do they just dissipate, or do they fall apart, or why are they not still there and still glowing?
They will still be there for a certain amount of time. That's a good question. And every protein is sort of different. And luciferase has been engineered so that it hangs around for a long time in the cell, and then some variants where it's highly destabilized. It's prone to unfolding. And once you get unfolding, the cell knows what to do then. It takes it through the proteasomal degradation pathway and has ways of breaking down different molecules. So it can kind of be, it's been engineered and developed to the extent where we kind of know how long it's sitting around. But it's degraded just like any other protein in the cell after it's, been around for a period of time, usually through the proteasomal degradation pathway. Good question. Okay, so once we have all of these reporter genes in place that we would like to use for studying, there's a whole bunch of brute force methods to actually get them into a cell of interest. So you can use different viruses, lentiviral, retroviral transduction. You'll probably come across these terms at some point in your career. You can hit them with some chemicals to burst open their membrane and let that gene come through. You can even shock them with electrodes to let that gene come in. And once you have that gene inside the cell, just simple application then of the luciferin small molecule produces the glowing cell. And that's, in a nutshell, how that works. Just a few comments about the actual substrates themselves, the small molecules. It's really critical that those molecules have access to our luciferase-labeled cells, right? They're the other part of the reaction. If they're not there, there's no light emission. Okay, so we need the luciferase to be transcribed, and luciferin has to be able to get to them. Remember, we had all these different kind of bioluminescent systems, and the one that's most often used is the one from the firefly, because it's that small molecule that once you inject it into, let's say, a small animal, it has a pretty long circulation time. It can get to almost any tissue, which means it can get to almost any luciferase-expressing cell, and that's really, really critical, even inside the brain. Whereas some of those small molecule substrates, like that cilantrine, that really weird-looking molecule that was used by the jellyfish, that doesn't have as, really like good properties for circulating everywhere in the body and having access to all different tissues. So it's just like the same thing that drug companies face, right? Pharmacokinetics, pharmacodynamics. Those concerns also apply to these molecules, but we got really lucky when we discovered that that small molecule had access to many different tissues. That's really lucky. Okay, so here's some of our first sort of bioluminescence images. When we took the firefly luciferase and put it onto one of these promoters, that just means it's on all the time. So we're just making luciferase all the time in these cells. Yeah?
Are the promoters actually genes, or are they
They're things that activate genes, but they're still made, with nucleotides and things. So they look just like a gene, but their actual purpose is to bind transcription factors and things in the cell to turn them on. So when we put these cells that had our luciferase gene just in a dish, and we dumped some luciferin on them, we could record their light emission using what's called a CCD camera, or a charge-coupled device. And remarkably, this number of cells in each of the wells is indicated at the top here. And we could see down to just a few cells in those wells, making luciferase and giving off light. Okay, and so this was our first clue that we might be able to detect some pretty sensitive processes inside animals, let alone just sitting here in this dish. So we had kind of went through just briefly how we were going to get our luciferin molecules inside of these cells, and we were able to do that using just some standard cloning techniques that we went through. And now our biggest challenge is, well, how are we going to actually detect that light emission within inside animals? We could see those cells sitting in a dish. But if we have luciferase expressing cells inside an actual living thing, we have all sorts of other challenges, right, if we're trying to detect that light emission. And that's because there's not a clear path from this light source to the detector in this case, right? Once you put light and put anything in front of that tissue, or put anything in front of that light source, the light's going to scatter, it's going to be absorbed, all these sorts of things which kind of lower our efficiency of collection. So there's all these sorts of things that we needed to consider in small animal imaging to actually make this useful. You know, how bright did the luciferase have to be for us to see it inside of a whole animal? Like, we're essentially burying it, you know, in the confines of a living organism. You know, how far down could it be before we lose seeing it? What about skin and fur and all those sorts of questions? And then the whole thing is curved, right, which kind of splays out our signal. So there are all these different challenges. What we were attempting to do first was, okay, it's pretty apparent if you take different colors of laser light and put them behind your fingers that the red laser light, showing you here, that comes through tissue much easier than either green colors or blue colors. And so if you take a green laser and hold it behind your finger, you get essentially nothing back, and you get nothing back when you use a blue light laser. If you just take a normal flashlight, right, your fingers always appear red. It's the red light that comes through skin most easily. And so that's why we thought we would probably have a better chance of seeing luminescence inside whole animals using our firefly system. Remember, that's the one that gave off the most, you know, kind of orangish red light. You know, those were longer wavelengths that could more easily penetrate the skin, and that was absolutely critical for us in the system. And it turns out if you kind of do this same experiment with a mouse, here's our mouse here, and just shine like white light at it, which is represented by this camera here, you'll notice something from the types of light that come through this animal. And all these different numbers on the back here correspond to the transmission of the type of light coming through these animals. And something that's very, very stark here is you'll see, like in all cases, they kind of go along with these lines. All of a sudden, there's a jump at 600 nanometers. There's like a big break at 600 nanometers. And also, the tissues that let the most light pass through them are the ones where there's not as much tissue to go through, right? So here's position number 1 versus number 12, just dealing with thicknesses and depth, okay? And the reason why there's all these issues with why different colors of light come through and why some are less efficient and some positions are more efficient. Probably the major trouble in imaging and why there's a big break at 600 nanometers is there's many, many different molecules that can absorb light in your tissue, and primarily your blood. Okay, so blood really, really hurts bioluminescence because one of the major components of your blood, what makes it that red color, hemoglobin. It's an iron-carrying porphyrin that hauls oxygen all around your system. It can absorb light of the same color that the luciferin system gives off, so that really kills our efficiency. And then there's all these other things, of course, in biological systems that wreak havoc, okay? Any organelle within inside your cell, mitochondria, nuclei, all these things, they can, scatter wavelengths of light, making it so that our camera can't see it from inside the animal. And I'm just going to show you that the hemoglobin absorption spectrum. So I think Marco said he took a lot of absorption spectrum recently, or looked at laser light scattering of plant tissue. If you did this with mouse tissue, you would notice that there's this big sort of absorption peak right here from either hemoglobin carrying oxygen or hemoglobin not. This is the red stuff in your blood. And it overlaps sort of significantly with the light given off by the different luciferase systems. Here's the one from the firefly. So here's the hemoglobin absorption spectrum in the background. And only light that's beyond 600 nanometers efficiently makes it out of the body. So that's why there was that big jump always at 600 nanometers on the mouse that was being hit with white light. Okay, so the more red we go, the better off we are. So that brings me to the question then, if we're going to attempt to use luciferase-labeled cells and bioluminescence inside animals, which mouse model do you think is best to use? Which one of these guys? Fat mouse, skinny mouse, hair, no hair, black hair versus white hair, brown hair? Yeah.
You want a thinner one just with more tissue there is, the less light gets through.
Yeah, that's great. So the thin, definitely the thin one. So that rules out this guy, the obese mouse. What else? Black fur versus white fur. Which color absorbs more light? Yeah, go ahead.
I don't know anymore.
Oh, it's okay.
I was just thinking that, like, whenever, like, black stripes and the white absorbs, or the other way around? No,
Other way
around. The black absorbs and the white reflects.
Mm-hmm. So in our case, if we're trying to detect light emission inside the animal, do we want that light to be more absorbed by the mouse skin or not?
Less.
Yeah, less. So that would say that, if we have to have fur, the white one's better. Okay, it's not going to suck up all the signal that we're trying to see. And then ultimately, it's the scrawny nude mouse, right? It doesn't have any color interference, right? No hair. The poor little guy, it's the skinniest. Okay, so a lot of times you'll see animal mouse models using this type of system just because the light is more easy to come out from it. And then if you have to make a second choice, then the white mouse is the next best choice, the one that has fur. So you'll see some examples of this after the break. I've just got a couple more slides and then we'll take a little break. So those were the considerations in terms of using the mouse model. Now the instrumentation that's actually used to collect luminescent light from these animals, I'm showing you here. These are sort of cool systems that have, they're nothing more than a glorified black box, right, to stick the animal in so that there's no stray light from outside coming into that animal. And they have a really, really, really sensitive camera on the top that's cooled with like liquid nitrogen and everything. So every photon coming out of that animal, every little piece of light coming out of there is going to be captured by the camera. And each one of these setups can house different numbers of animals as opposed to maybe 300,000, let's say, and the most expensive component is just going to be that camera, okay, just to detect all photons of light coming out of it. So in one of those setups, then, once we have an animal that has any sort of luciferase-expressing cells in it, we stick it in one of those black box cameras. What we read out then is our bioluminescent signal from one of those cooled CCD cameras at the top, and then the output just overlays these two using just a normal photograph as a reference image. And then we can watch then these changes in light emission and where that signal is at over time. So that's how the instrument actually reads out the signal. And corresponding to that plate of cells we looked at earlier, just to see how many cells at a given time we can see with one of these systems, we saw it all the way down to about 1 or 2 cells in the dish. But when we moved to animals and all those challenges we talked about before with skin and fur and blood and all that stuff, we're more in the range of about 100 cells that we can reliably pick up. And these are coming from luciferase-labeled cancer cells just implanted into the abdomen of these animals. And we are trying to determine just what our lower limit of sensitivity was inside those animals. And of course, because of all those absorption and scattering issues, the number of cells we were able to detect using the in vivo model was a few orders of magnitude lower than what was in the dish. And I am going to stop right here before we talk about, after the break, what exactly we can do now that we have all of these things in place, how we get luciferase and luciferin into those cells, which animal model to use, what the system is to collect the light, and now what are we going to do with it. Okay, let's take a break. Again, this is the coolest part of, I think, what I get to talk about other than the nuts and bolts that we went over sort of for the first part. I'm going to talk to you about some of the things that I personally work on using bioluminescence as a tool then for tracking biological processes, and then what some of my colleagues in Chris Contag's lab have been working on throughout the years. So this is going to be where bioluminescence, I think, comes to light for how it can translate and actually impact human health. So we'll start by looking at a few areas where there's been an impact in our understanding of different physiological processes, and then what remains to be done in bioluminescence. The field is sort of wide open, in my opinion. And just to come back to our routine figure here of how this whole process works, and every case study I'm going to show you this far, we use the same techniques to introduce luciferase and luciferin into our cells or molecules under study. We use small animal models as our host organisms, and then we use our cooled CCD cameras to read out where the light is originating from in those animals. So in our first basic example, there's been a lot of work done in cancer imaging using bioluminescence to simply understand how cancer cells move about, how they migrate, how they respond to therapy. And so in this particular example, I'm going to show you breast cancer cells labeled with the luciferase from the firefly using the genetic techniques we talked about earlier. And in almost all the cases, the promoter that we're using is something that keeps the luciferase turned on all the time. So they're constantly glowing in the presence of luciferin, making it easy to track. And in most of my work and my colleagues' work at Stanford, we've been using these cells to look at the metastatic process. And so we originally implant those cells into this region here of the animal, which is a fat pad where breast cells like to grow very nicely. But over time, you'll notice we have this signal accumulating at the lung. Okay? Breast cancer cells often metastasize to lung and neural tissue. And just to underscore how this bioluminescence is a good readout, when you actually dissect the mouse and you see all these little white globs in there, those are the tumor cells. Okay, so we're actually seeing at this stage the metastatic process in the live animal, and that becomes really useful for us then to evaluate either therapies that block that cascade and understand the actual genes or molecules involved in the metastatic cascade. And this sort of approach has been used to study not only breast cancer, but almost every type of cancer you can think of. Different cell lines have been labeled with luciferase, including those from the colon, the brain, liver, etc., and studied in a very similar fashion. And that's the subject of ongoing research all over the country. In of particular interest, I think, to many of us at Stanford, since we have access to the clinic and the hospital right next door to our research lab, we want to use this model not just to understand the trafficking patterns of these cells, but how they actually respond to therapy. And at what point do they respond to the therapy? The bioluminescence readout gives us sort of a window into what's going on inside those animals in real time. So we took some of those breast-labeled cancer cells that on a previous slide, implanted them into the mouse again, and then evaluated their growth in the presence of some common chemotherapies, 5-FU and cisplatin, which are still used in the clinic, and other things such as adriamycin, doxorubicin, all these common chemotherapies that you may or may have not heard about. We've studied in this fashion, using bioluminescence to understand if they're effective at all. So, in the experiment, what we're looking for, using bioluminescence of a readout of growth, right? The more signal we see over time, that indicates the cancer cells are still proliferating, dividing, and growing, and the therapy is not effective. However, if we see that signal die off or remain stable, that should mean that the chemotherapeutic is having an effect. And so, in our animals that were treated with just like a placebo, sham, nothing. PBS is similar to water. But in those animals that were given the chemotherapeutic, we noticed that in this 5-FU case, we have maybe more of a delayed response before we see this huge outburst of signal, meaning that the cancer is spread everywhere throughout that animal. Okay. And then in the case of cisplatin, we seem to have more of a general effect against this particular type of breast cancer, in that at least in 2 out of the 3 animals I'm showing you here, we have no spread of the cancer throughout that body. Okay, so this is how bioluminescence in a general sense has been used to look at chemotherapies and chemotherapeutics. In some of the work I'm personally involved with at Stanford, we've been using bioluminescence to help us gain insight into kind of a whole new class of therapeutics, those that aren't based on chemicals like cisplatin and those structures that I showed you, but those that are actually based on a patient's own immune cells, the cells that should be there to fend off disease and might, hold tumors at bay at an early stage. But in a cancer patient at the clinic that comes in with actual tumor mass, at this point, their immune cells, which I'm showing you here in green, can no longer control, the outgrowth of the tumor. But there's a general feeling that if we can take those immune cells out of that patient's body, give them an extra boost, so that then they can be reinfused into that patient and then go on to target and destroy this tumor. This is a class of therapies known as immunotherapies or cell-based therapies that has gotten a lot of attention in the press. And so we thought that we could study the effect of that particular immunotherapy using a class of immune cells, which at Stanford we call them cytokine-induced killer cells, or CIK cells for short. And they just come straight from human patients. We give them an extra boost of another immunostimulant to give them some more fighting power. Then we inject them back into the patient in the clinic, or in our case, we're going to evaluate how well they do in an animal tumor model. And so in this case, we're using luciferase-labeled cervical cancer cells implanted in the animal, and we're treating them with these potent CIK cells derived from the mouse. And we kind of, at least in the case of cervical cancer, got a varied output. Again, we're tracking the luminescence of the cancer cells over time. If the therapy is effective, we should see no change in luminescence signal over time. There should not be an increase. However, in some of our animals, we see this kind of big burst of light emission coming from the body, indicating that the cells were not effective at that stage in the animal. And so this was telling us, okay, well, these cells seem to work in some cases and arrive at tumor tissue, but they weren't always effective. Is there some other way that we could engineer them? And so what we've been working on now is taking studies and labeled with luciferase and looking at tumor outgrowth over time. We wanted to see, were these cells, actually arriving at the tumor site? If they didn't arrive at the tumor site, that would explain why they weren't, effective at decreasing or wiping out the tumor. So we just kind of reversed the experiment, put the luciferase into the CIK cells, injected those cells into tumor-bearing animals, and then gave the animals some luciferin to make them glow, and we did see, at least over time, that these cells could accumulate at the tumor site. Okay, so that was very encouraging. So what we're exploring now is, since these cells are very effective at arriving at different types of tumors, could we load them up with other drugs, or in this case, an oncolytic virus that's very tumor-specific and very potent, and use them sort of as a delivery vehicle. And that work is ongoing, both in clinical trials and then in mouse models within our own research lab. All right. In all these cases of labeling tumor cells or immune cells and evaluating their consequences, we've been using our promoter luciferase constructs, putting them into the cells, and then transferring those cells into the mouse model. Now this becomes really, really tricky if we want to study a very few number of cells, or cells that are really hard to isolate, to carry them through this process. Like, what about a stem cell? It's hard to get your hands on a large quantity of those and, blast them with electricity or something. They're not going to survive that. Okay, so what we've done instead is we've taken a different approach to get around that problem. And that is we engineered mice to express luciferase in every single cell of their body, so that whatever cell type we wanted to study, then we could just go into that animal and get the cell. So we actually made what's called a transgenic animal, where now every single cell in these particular mice is expressing some amount of luciferase. And I'm just going to show you that, of how we were able to verify that all the tissues are expressing luciferase. As if we inject luciferin into them, they should just start glowing everywhere. Okay, so once, I'll show you here. Once luciferin is put into the IP cavity, you'll see the molecules diffusing around. This is an image acquired over about 20 minutes. We'll play that again. You'll notice the brightest spot is, if I can get it to play again. The brightest spot was right over the area where it was injected. And then the molecule slowly diffuses out of that area and around the rest of the body. Maybe I can't get it to play again. There we go. Okay, let's do it again. Okay, so here we go. Right where the edge of that needle is, you'll see the most intense signal being given off in the intraperitoneal cavity, is what IP stands for. It's just right in the space near the abdomen. And then similarly, we also injected that small molecule luciferin into the tail vein of these animals, so like directly into the bloodstream, just like when you get an IV at the hospital and they stick that thing right into one of your veins. That's what we did with the luciferin substrate here. And you'll notice it goes much faster than when it was just sitting in the abdomen. That's because once we've already put it in circulation, it's whipping around the bloodstream of that animal very quickly. But these were kind of just images to verify that, okay, yes, we'd actually engineered a mouse so that every tissue, every cell was expressing luciferase, so that we could then use these to study other processes, stem cells, and very hard to isolate cells that we may want to track around. I'm going to show you one example of that.
I have a question.
Yep.
Is that mouse going to just glow forever, or is it
It's a great question. Luciferin does get used up, and you kind of saw at the end of those images how the signal trails away. So there's some tricks you can play if you want the mouse, let's just say, glow forever. You can actually build a pump that delivers a certain amount of luciferin all the time to that animal. Or you can simply inject more, or kind of like formulate the luciferin into something like aspirin would have slow release properties. So there's all these sorts of cool tricks you can play to prolong your signal if necessary, but it will go away over time. Within these transgenic animals, though, that we had created with luciferase in every single cell, I'm showing you here just a collection of cells that we took out of those animals just to see if all of these cells were glowing, if they had light emitting properties. And they did, all to a certain extent. I'm showing you them just sitting in the dish again and dumping luciferin on them, and then we're recording the light emission. And the actual light emission is represented by these bars here and just visually within the middle well. They all had different extents of luciferase protein in them, but they were glowing enough that we thought we could use them to study certain things like bone marrow transplants. Now, this was kind of born out of some of interest in the hospital at Stanford, where quite a few bone marrow transplants are done. They're one of the leading communities in both bone marrow transplants for leukemia patients, and then for other people requiring that after chemotherapy. So, oddly enough, within all of our bones, right, there's all these different blood-forming cells. It seems kind of weird, but they're just a factory of all different kinds of white blood cells. And some of the cells I showed you that were emitting light on that previous screen. And we thought that we could gain insight into the whole process of bone marrow transplantation and when it can go wrong. There's all these stories of you need to be a certain match, right, to donate bone marrow for a person that would need new blood or a leukemia patient. And so we thought we could understand this process more of how things are rejected by our bodies or what types of bone marrow are okay using our small animal model system. So what we did is we took that transgenic mouse where all those cells were expressing luciferase, and we cracked open the bones from one animal and harvested all of the cells that were inside that cavity. And then in a transplantation into another mouse, we could watch then the formation of the blood system over time. So exactly what happens in a bone marrow transplant. You're trying to replace unhealthy blood with healthy new blood. And these here are a series of the bioluminescence images taken from the animals then that were given both the bone marrow transplant and the small molecule luciferin. And just after a day, you can start to see what's likely signal coming from the spleen, another organ that has a lot of immune cells. Then over time, you start to see like this signal coming out everywhere from the animal, showing that we're actually regrowing blood within inside these animals. I found that to be a pretty remarkable feature. And so the researcher involved in this has now been using this particular system to understand, how closely matched does the donor and the host have to be for the blood system to grow out, and defining a real issue and bringing some clarity to a real problem in the clinic in terms of when is it okay to transplant, when is it not. And just to delve in a little bit deeper with the transplantation system, all those blood-forming cells that were coming out of the bone, they actually derive from one cell, a hematopoietic stem cell, or HSC. So like all stem cells, it has that cool capacity to make more of itself and then differentiate into every other type of cell out there. And it's actually this cell in the bone marrow that is critical to that process of growing out, reconstituting blood. And so when my lab mate Yuan took a look at this process, not just using all the components from the bone, but could we actually track a process using a single hematopoietic stem cell, just one of those cells. He isolated it, looked at it in a 96-well dish before transplant, verified, yes, there's one cell under a microscope here. Since it came from our transgenic animal, it already has luciferase. We did single cell transplantation into this recipient and then provided that animal with luciferin. We could see over time then that remarkable process of blood regeneration inside these animals from a single cell. And I should mention that in all these cases, the donor mouse, we've irradiated it and wiped out all of its capacity to regrow its own blood like normal. So the only chance that it has to survive is if this particular cell does its job and regrows the entire blood system. Yeah.
The donor, the host, has been irradiated.
The host, sorry, did I say that backwards? Yeah. This mouse here, the host. And what was interesting is that it seemed to vary between animals of where we had initially see a focal spot and then this huge explosion. In some animals, we saw it, in near the joints of the legs, but it was kind of random throughout the bodies of different animals. And I think that was kind of maybe what was most surprising to the field. It wasn't an absolute process of where this was taking place, but kind of wherever that cell home to first found a home and then grew out, that led to the formation of the entire blood system again. Some of the other areas, I just kind of went through basically a lot of work on how we've used bioluminescence for cancer cells, immune cells, stem cells. It's also had a big impact in looking at not only chemotherapeutics, but evaluating novel types of therapies aside from bone marrow transplantation. One of them being gene therapy. You might have heard from the news some instances about areas where transplanting new genes into a particular cell and into a host would be therapeutic and beneficial. And probably a classic example of this is in the case of cystic fibrosis, where a bunch of fibrils build up near lung tissue preventing easy breathing and causing mortality in a number of people at a very young age. And this has all been traced back to a defect in one particular gene known as the cystic fibrosis transmembrane receptor. And the whole thought behind gene therapy is if we could just repair that gene, get those mutations out of there, we would have cured this disease. So there's been a number of different approaches taken to try to how do you get a new gene delivered into a diseased tissue, and bioluminescence has had a big impact in this area. And I'm going to show you one example where we've used it to figure out how, what kind of conditions we can use to introduce genes using viruses, which are entities that can really infect host tissue quite well. And the whole thought behind using viruses is that they're really good at recognizing human cells and then injecting all of their insides into a particular host. Could we use them to deliver them, a new gene? And the kind of the general schematic of that is here, where let's say this is the gene of interest that we want to introduce for cystic fibrosis, the repaired gene. Can we generate some virus that has that gene in place, inject it into our host, have it infect those cells that are that have the copies of the wrong gene, and then cure that patient. So the way bioluminescence has impacted this area is that we have a very easy readout. If we use firefly luciferase as a surrogate gene, we can see where it's at, where it's been infected, and to see what sorts of delivery vehicles, like the virus or some of those other methods that were on that first screen, if they're going to be effective at all in this approach. And so we've been using bioluminescence technology to introduce the firefly luciferase gene as kind of a model system. And then whatever cells that it hits, we give the animal luciferin, we should be able to see exactly where that gene was incorporated. And in this case, the viral vector that we're using is an adenovirus. And to target things like cystic fibrosis, where the mutated or the incorrect gene is in the lung tissue, we thought, well, we'll have these organisms, or we'll have the mouse kind of breathe in our luciferase-labeled virus. And if it was effective at all in introducing our luciferase gene into lung tissue, we'd be able to see bioluminescent signal coming from the lung. And that's what we found in some of these animals that had inhaled our luciferase-containing virus. You see that bioluminescent signal coming from the lung tissue. And if you actually dissect this tissue out and dump some more luciferin on that lung, you'll see the little pockets of where the virus was actually able to deliver our gene. Because it's a good model system for thinking about, well, what other kind of genes can we introduce in this manner. The problem that we discovered was that while we could see quite a bit of bioluminescent signal coming from these animals, it didn't persist for very long, or it didn't seem to be that we could deliver a lot of the virus to portions of the lung that were away from the vasculature or deep into the tissue. And so we used our animal models and our virus and luciferase vectors in combination with other sorts of drugs to have a more prolonged release of our virus. And so we use bioluminescence in this case as a readout of the effectiveness of some of these combination approaches. And I'm showing you here different examples of our luciferase-labeled virus in combination with perfluorocarbons, which is just a nice sort of packaging vector for the virus itself, permitting slow release of the virus. And our hope was a more prolonged incorporation of the firefly gene in our model system. And what we found was that if we did incorporate one of our viruses labeled with luciferase in combination with the perfluorocarbon, that we could see this prolonged level of luminescence inside the animals. And so bioluminescence enabled us to get a handle on what sorts of conditions might work well in the context of gene therapy. The last area of research that I wanted to touch on of what we've used bioluminescence for down at Stanford is in the area of infectious disease. And this has been sort of a fun area, I think, to study because bacteria, they're really smart. And unlike all the systems that we talked about before with firefly luciferase and using those to track cancer cells, we always had to inject in the luciferin, right? So we either had to inject it by pee or in the tail vein, but we needed to have a source of the small molecule. The bacteria, they know how to make their own luciferin, okay? So if we have the bacteria labeled with one particular long gene, the lux operon, in that gene is encoded all the enzymes needed to make the bacterial luciferase and the small molecule itself. So once we get that gene introduced into a particular bacterium, it's always glowing. No addition of the substrate required. The only downside, of course, is that in all these cases, the light emission coming off is blue. And what we had talked about before, the most attractive feature of all the other luciferases, the firefly one, was that it had more red light associated with it, so it could more easily penetrate the skin. So there's kind of a little trade-off between these systems. But all that's necessary to monitor bacterial infections in your favorite bacterium of choice is to introduce again that whole big long chunk of DNA encoding the luciferase and the luciferin into your bacterium, then they'll just glow continuously. So that's shown quite elegantly in this model of kidney infection of this particular bacterium. You see here, you can already see it glowing in the syringe, and we're using a catheter here to deliver it into the animals. And over time, these bacteria colonize the kidney. Okay? And we've been using this particular model to look at different antibiotics and to track those bugs over time inside the animal, but no addition of substrate required. The other major area of research in our lab involves the bacterium Listeria monocytogenes. And this is a pathogen that's associated with a lot of food poisoning incidents, especially from potatoes. And you kind of hear about it in the news every so often. I think there was just a couple of weekends ago another incident in Minnesota with some listeria-infected potatoes. And so there was interest in our lab in studying this particular bacterium, and so we introduced the bacterial luciferase substrate system inside that bug, and we implanted it into animals as a model of infection. We found for the longest time this persistent signal coming from the mid-region of the animal. And we had no idea what that was until we kind of did a careful dissection and figured out that it was actually the gallbladder from these animals. And the bacteria, which are these kind of long blue stained things here, were just sitting within the gallbladder, lined up on some really mucus-type structures. And that was such a shock to the community because the gallbladder, it stores all of those enzymes and salt that break down things in the liver, right? It's like the nastiest place you could ever think of to live, and here these bugs were hanging out in there. So this has spawned a lot of research into, do listeria hang out, in the human gallbladder? What's going on with that? So I think bioluminescence sort of opened up our eyes to some interesting pathology with this particular bacterium. And similar to the cancer field, we've been using bioluminescence to look at different drugs to treat bacterial infections using luciferase labeled, in this case, Pseudomonas. And these animals up here have the bacterium associated with a wound on their backs, and then we've been investigating different antibiotics and their effect on killing off those bacteria over time. Okay, so becoming sort of, again, like a discovery tool for a quick readout of therapeutic efficacy and what does or does not work for antibiotic treatments. And I think one really exciting area of research that remains totally unexplored at this point is that this big gene that's been used to introduce the luciferase enzymes and substrate into bacteria actually, in several different bugs, is what is turned on and off in response to various small molecules. It's like a little chemical code of communication among bacterium. And in the last 5-10 years, it's become apparent that bacteria don't really live in isolation, but they sense each other using a whole host of different small molecules to sense just how many of my buddy are in the area so that we can launch an infection. And many, many of these small molecules that bacteria use to communicate amongst each other are tied into a luminescent readout. So the luminescence can become a really good, I think, discovery tool for looking at all of these different small molecules that bacteria are using to talk amongst one another before launching into an infection. Okay, so that's kind of a smattering of the work going on down at Stanford, and then I'll just, in the last few minutes, highlight where I think some of this work is going, and some things that might interest you as far as future research projects. And I've highlighted some of the problems with bioluminescence or some of its limitations at this point earlier in the talk. One of them being, remember, we always had that 600 nanometer cutoff for light emission because hemoglobin in the blood kept absorbing a lot of the light coming out of these luciferin systems. It'd be really nice if we had an enzyme substrate pair that emitted light outside of where that hemoglobin absorption curve was. That emitted light around 700 or 750 and was a red emitting fish, like that was mentioned earlier on. Fish produce red bioluminescence. And there's one example, the black dragonfish, that can emit red light. So wouldn't it be cool if we could figure out what's the enzyme luciferase and what does that small molecule look like, and then transition that system for use in small animal research. Other people have tried to come up with red-shifted luciferin systems just using mutagenesis and cloning techniques, just by brute force methods, taking our gene for luciferase, introducing all sorts of mutations into it, seeing what comes out. If you focus just on this panel here, one group of researchers very recently swapped out different amino acids within the protein, and what they ended up producing was, here's our normal wild-type firefly luciferase, yellowish, greenish in color. They were able to produce variants that were more red-shifted. That becomes really useful for tracking single cells in vivo. So I think there's still some more optimization that can be done in terms of looking at how can we improve our light detection. And one obvious question, I think, is can we actually engineer that bacterial system into mammalian cells? We don't have to keep injecting luciferin into our animal models to look at cancer cells and so forth. And I think there's been some attempts to take that big, long construct, that the bacteria have figured out how to encode all the things in one pot and move it into, a human-type cell. But there's some expression level issues and things that haven't been worked out yet. Another very big, important, lingering question is, where does the normal luciferin from the firefly that we use in all of our animal studies, the mammalian cells that we keep injecting in, where does that come from? Can we figure out how to biosynthetically make it and then genetically encode it so that we have the mammalian cell equivalent of the bacterial system? I think this would be really, really cool to figure out. How is that made in nature? Where is the firefly getting that from? Are they eating it? Is some bacterium producing it? Or do they make it themselves? Still an unknown question. And then, another cool thing is that the firefly is thought to only be born with a set amount of luciferin. Okay? So that it doesn't acquire it any, at other points in its life, which kind of rules out it ingesting it. So if that's the case, once that molecule, undergoes the bioluminescence reaction, it has to somehow, recycle that small molecule. How does that happen? And if we can do that, can we prolong our bioluminescence signal. Yeah, you had a question?
How did they think that it was originally making luciferin in the first place before it was created?
Sorry, say that again?
Well, do they know where luciferin comes from? What it's like for?
Yeah, it's not known if it's coming from a bacterium or a biosynthetic pathway inside that animal. The only thing that's been charted is luciferin content over time, which is standard chemical-based approaches like HPLC. I think these are some fundamental, cool questions to think about from a biotechnology point of view to improve bioluminescence imaging and its ease of use in the future. From an engineering point of view, a big question in bioluminescence has always been, what's the depth of the signal? All of these images that we've been looking at for bioluminescence have that light superimposed in a two-dimensional fashion on the reference image. But how deep is that signal, right? Is this a half a centimeter in, a full centimeter in? We don't know. Because right now, the only information that we have is being collected from one angle. And you can imagine that to get depth information, if you just collect light all the way around the animal, you'd be able to more quickly localize where three-dimensionally that particular source of signal is located. And those are types of instruments that are being developed, and here's a prototype is such an instrument where you have a three-dimensional rotation about the animal of interest. And then probably in line with some of the other speakers you've heard thus far, the bioluminescence is related to other types of imaging techniques, fluorescence, nuclear type approaches, such as PET and SPECT, and this same reporter gene technology can be used in that fashion. So instead of having our promoter only producing luciferase, it could produce luciferase, plus GFP or another fluorescence protein. And that way we can look at many different imaging techniques all at one time, which is where this multimodality term comes from. So there's a lot of new interest in multimodality molecular imaging. I'm going to skip over a couple of these to a main question I think that always comes up with, does bioluminescence have human clinical utility? And because all the data I've shown you or all the pictures I've shown you have always been from the mouse. And I think the answer is maybe, because we're always limited by depth, right? And how hard it is to get light out of things that are thick. So we're much fatter than a mouse, but there is some work being done and using bioluminescence to look at things that are really near the surface that you can put right up against a camera, things like melanoma or skin cancer, and perhaps looking at changes in the GI tract with bioluminescent bacteria. And then, finally, other work that I've done has taken human cancer cells straight out of the human patient, not just from cell lines like I've shown you before, but straight from a tissue biopsy and into an animal to evaluate whether or not certain therapies are effective against that particular patient. And that's an example of what I guess has been coined personalized medicine. Okay, so just to summarize just briefly here with our scheme of using luciferase-labeled cells and viral vectors and small animal models, how we're able to use bioluminescence imaging to track disease, to evaluate different therapies, and then to look at things such as bacterial infections. And probably the one thing I want you to take away from this talk today is how bioluminescence can facilitate biomedical research and discovery, even if it isn't currently used, let's say, in humans. And that's because it allows us to visualize where cells are at, how they're changing dynamically within the context of actual living tissue, so that we can gain insight into different processes and steps of disease that were previously unknown to researchers. And I just want to acknowledge a couple of people who made all this work possible. Chris Conte is in charge of this lab at Stanford where most of this work is taking place. And these are several of my lab mates whose different work I was highlighting within this talk. And then in the background here, you can see this kind of space-age looking building. That's actually where the Contag lab is housed, right across the street from the hospital at Stanford. So I'll take any other questions that you have for me. It was fun to be back.
Question? Well, let's thank a speaker for a very